Recording right now, and thank you very much for agreeing to give the talk, Drew, and it's all yours.
Thank you. My name is Drew Tack. I am a research biologist at the National Institute of Standards and Technology, or NIST, in the Cellular Engineering Group. And I'm going to be talking today about some of the work that we've been doing in engineering biological function. So before we get started, I want to start with showing this preprint on bioRxiv, the genotype-phenotype landscape of an allosteric protein. Most of the information I will be presenting today is in this, this preprint manuscript, and we are actively recruiting postdocs. So if any of this sounds interesting to you or to someone that you know, please feel free to get in contact with me and we can start talking about it. Okay, so our ability to engineer biology is rapidly expanding. Advances in gene synthesis and design overcome the chemical limitations that have held us back in the past, and directed evolution and other approaches are capable of exploring the complex landscapes that biology lives on. These new methods can engineer functions from things as small as simple proteins up to entire cells. But assembling biological parts into complex systems rarely results in the function that we intend. Instead, we have to build, test, characterize, troubleshoot, and then adjust our workflows to accomplish what we're hoping to. In order to reach a point in biology where we can assemble parts more like a puzzle with pieces that fit together and result in a final product that does exactly what we want it to, we need to reevaluate how we construct and build biological parts. And at the foundation, transitioning biology from a qualitative art to a quantitative discipline will improve the results in many endeavors that involve biology. To fulfill the full promise of biology, we really do need to establish quantitative methods for assembling biology. So today I'm going to talk about transitioning the engineering of allosteric transcription factors or genetic sensors from a qualitative process to a quantitative discipline. Allosteric transcription factors are very important in biology. They allow cells to regulate gene expression in response to ligands, so they can adjust to dynamic environments that they encounter. For those of us who are interested in building biological constructs with new purposes, genetic sensors are important because they allow us to program complex cellular functions into living cells. And they're critical for implementing many of the proposed applications of engineering or synthetic biology. In a simplified summary, genetic sensors are allosteric proteins that exist in two distinct conformations: a DNA binding conformation and a non-binding conformation. The transition between these two conformations is thermodynamically driven by the absence or presence of a ligand. By far the most common example of an allosteric genetic sensor is the Lacl repressor from prokaryotes. It is used nearly ubiquitously in many different fields of biology to control gene expression. Now, the conformational switching process of genetic sensors is modeled and parameterized by the Hill equation, shown here. For Lacl, in low ligand concentrations, the sensor is in the DNA binding conformation and gene expression is low, and this is parameterized by G0. At higher or saturating ligand concentrations, the genetic sensor enters a non-binding conformation where gene expression reaches some maximum. This is parameterized by G infinity. The concentration of ligand at which this conformational switch occurs is described as the effective concentration and is represented by the parameter EC50. There is a fourth parameter, N, which describes the steepness of this response. I acknowledge that it exists, but we won't be talking about it today. Gene regulation by Lacl is one of the most studied systems in biology. Because of its general usefulness, there have been many efforts to qualitatively improve or change Lacl. For example, people have engineered Lacl variants with decreased EC50, making the sensor more sensitive to ligand, or they've decreased to the G0, reducing the leakiness of the sensor. They've also increased G infinity, which is improving the ligand response, and some efforts have even inverted the dose response curve entirely. Now, generally, these changes are considered desirable, but for building biological constructs to solve and address real-world problems, we don't need qualitative improvements on sensors. We need sensors that have and fill exact and existing needs. We are great at generating diversity. The challenge really comes in characterizing that diversity, both at the genotype and phenotype level. Directed evolution remains the most popular method of engineering biology, where qualitative selections, positive and negative, qualitatively whittle down the diversity in a library to some subset with desirable features. A set of those surviving genotypes are then quantitatively characterized, and sensors that meet the desired responses are generally selected. While directed evolution is certainly capable of identifying sensors with improved functions, as I have just showed on the last slide, we need methods to engineer genetic sensors with quantitatively prescribed functional parameters. We imagined an ideal solution would be to simply measure everything. And that includes at the genotype and the phenotype level. If we could measure everything within a library of genetic sensors, then we could simply select sensors that have the exact functions that we need for our application. This would allow for the quantitative engineering of genetic sensors that would exactly match the needs that we have. And fortunately, technologies in sequencing, design, synthesis, and robotics are all converging on a point where we can measure the entire genotype and phenotype of a library. So what does it take to measure the dose response curve for an entire library of genetic sensors? It took a lot of troubleshooting, optimization, automation, and the implementation of DNA barcodes. DNA barcodes were central to this whole process. The overall workflow is shown here. But DNA barcodes first allowed us to characterize the entire genotype of the library by mapping the individual substitutions in each sensor to a unique DNA barcode. Then, by connecting the growth rate of each sensor, or by connecting, the growth rate of a cell to the function of the sensor, we could monitor changes in the relative abundance of barcodes to determine the phenotype of each sensor in the library. This was and is overall a pretty complex process. We're going to walk through it, and then I will tell you about some of the things that we can do because of this. In total, we had to design and build 2 different plasmids for this work. The first, shown here on the left, Lacl regulates the expression of yellow fluorescent protein, or YFP. In this case, the output of the plasmid and the genetic sensor is fluorescence, and this is a widely accepted method to measure gene expression and to characterize the dose response curve of genetic sensors. We could measure the dose response curves of a sensor in this plasmid using flow cytometry, which would give us measurements in molecules of equivalent fluorophore, which is a traceable unit and thus very meaningful. In the other plasmid we had, Lacl regulates the expression of the tetracycline resistance gene, TET-A. With this plasmid, the growth rate of a cell should be dependent on the activity or function of the sensor, and thus it should be directly related to the concentration of a ligand. The ligand we used was IPTG, and in order to use growth rate as a reliable indicator of the output of a genetic sensor, we had to make sure that the growth rate actually corresponded to the dose response curve of the sensor. And when we, after a little bit of optimization, when we measured the dose response curves of Lacl in both of these plasmids, we found that they were very similar, although with growth rate as the output of this sensor, and then fluorescence is the output of this fluorescence plasmid. So here we see the relationship between fluorescence in this first plasmid and growth rate in the second plasmid. We measure the dose response curves for several different Lacl variants in a range of different ligand concentrations to build this curve. And you can see that while the relationship is not strictly linear, they are tightly related. And from this relationship, we constructed a calibration curve between growth rate and fluorescence. With that established, the next step was to build a library of genetic sensors. So we set out to build a library of genetic sensors where we could measure the genotype and phenotype of every sensor in the library. We use mutagenic PCR across the entire Lacl gene, and randomizing positions across the entire gene eventually allowed us to identify positions within Lacl that had specific effects on the function of the protein. Now, during the mutagenic PCR process, we attached DNA barcodes to each sensor in the library. This library of sensors and their attached DNA barcodes were then cloned into the library plasmid, where the sensors regulated the growth rate of the cell by controlling the expression of the tetracycline resistance gene. We selectively reduced the size of the library to 10 to the 5th variants, which allowed us to determine the exact genotype of every sensor in the library using long read or PacBio sequencing. And in total, we sequenced a little more than 60,000 plasmids in the library. And this included the DNA barcode, the sensor, and any mutations that accidentally occurred in the plasmid backbone during the library construction process. And here is a breakdown of that sequencing information as it relates specifically to the sensor library. Most of the plasmids within the library were sequenced at least 3 times. With some plasmids sequenced 50 times, and one exceptional example was sequenced over 5,000 times. This sequencing coverage gave us a view of the genotypes that existed within the library in terms of the Lacl sensors. When we looked at the sensor function, we found that amino acid sequence was responsible for the measurable phenotype of a sensor. So we conducted all analysis in the amino acid space. And you can see that the most sensors in the library had between 4 and 6 amino acid substitutions with a statistical mode at about 5 substitutions per sensor. And overall in the library, 94% of the accessible amino acid substitutions appeared at least once. Now, the other important genotypic feature on the plasmid library is the DNA barcode, which compresses the genotype information. The DNA barcodes contained 54 positions with randomized nucleotides. And during sequencing of the library plasmids, we found that all 54 of these positions were pretty evenly distributed across the 4 possible nucleotides. This was important to ensure that each sensor in the library was attached to a unique DNA barcode. Additionally, throughout the barcodes, you can see interspersed constant A and Ts. These were put in to prevent the formation of restriction enzyme cut sites on accident, and they helped us identify these DNA barcodes in downstream sequencing analysis. Now, at this point, we know the genotype of the library. We fully sequenced every plasmid in the library, and we know the exact amino acid sequence of each sensor in the library, as well as the corresponding DNA barcode attached to that sensor. Now, sequencing the DNA barcode of a plasmid can tell us the exact sensor on the plasmid. And this DNA barcode is small enough to sequence with short read deep sequencing technologies. In particular, we use the Illumina platform. And these types of deep sequencing can measure the relative abundance of sequences in a population. And this happens to be very useful for characterizing the phenotype of each sensor in the library. Changes in the relative abundance of barcodes over time can indicate the growth rate of a cell. Now remember, the growth rate of the cell is tied to the function of the sensor, since the sensor regulates the expression of the tetracycline resistance gene. So the growth rate is a result of the sensor function at any given ligand concentration. We tested using barcode sequencing as a measure of cell growth rate by comparing growth rates determined from clonal growth curves in plate readers. So these are OD measurements or absorbance measurements over time. And we fit these to exponential growth fits. And then we compared that to the growth rates that we determined by measuring changes in the relative abundance of DNA barcodes at different times throughout this growth. And we found that the correlation between these two methods was pretty strong. Additionally, the real benefit here is that barcode sequencing allows us to measure the growth rate of many cells simultaneously in a single pot. And because of this strong correlation between the two measurements, we knew that we could use barcode sequencing as a reliable metric to calculate the growth rate of cells in the library. All right, we use the growth rates of cells measured with barcode sequencing to characterize the dose response curve for each sensor in the library. Characterizing the dose response curve of a sensor requires measuring gene expression at several different ligand concentrations. We grew the full library in a total of 12 different ligand concentrations, and each in the presence or absence of tetracycline. In total, this was 24 unique chemical environments, all contained within a single 96-well plate and maintained in exponential growth by periodic dilution. During growth, culture samples from the 24 different chemical environments were extracted or taken and prepared for short-read sequencing. The DNA barcode populations were sequenced at 4 different time points in total. And to facilitate the process of all 24 of these growth conditions, as well as samples for DNA sequencing, we used an automated laboratory system, the Living Measurement Systems Foundry, part of our cellular engineering group at NIST. This handled all of the growth conditions, or setting up all of the growth conditions. It has integrated plate readers that incubated the cells during growth, and it has an arm integrated to handle all plate movements and manipulations. From all of this, we collected a total of 6 million different data points that combined to give us about 1.5 million gene expression estimates, and in turn collapsed to characterize 60,000 dose response curves. All right. Barcode sequencing, as it was growing in these 24 different chemical environments, gave us 24 different growth rates associated with each sensor. An example of the growth rate data is seen here. We then used that calibration curve that I mentioned earlier to estimate the gene expression associated with each one of these growth rates. And these estimates were given in molecules of equivalent fluorophore, again, that traceable unit of fluorescence. And the estimated gene output data for each sensor was fit to the Hill equation, which is shown this black line of this curve on the right. And this parameterized the function of every sensor within the library. This was repeated for all 62,000 sensors that are in the library. So across the 62,000 dose response curves that we measured, we found a wide range of parameterized function. Sensors in the library had EC50s ranging from about 1 micromolar to about 2 millimolar. That's shown on these x-axis of these top 2 plots. Gene expression ranged from close to the background level of the flow cytometer to about twice the maximum gene expression of the wild type sensor. So overall, we had a wide range of functions to choose from within this library. We wanted to know if we could engineer genetic sensors with specific functions by choosing sensors from the library that met exact targeted functions. To examine this, we targeted sensors that had specific EC50s and G-infinity parameters. Now, as a reminder, at this point, all we have is growth rate information. We've computationally estimated the gene expression of each of these sensors. But in order to verify that these estimated gene expression outputs correspond to the actual function of the sensor, we had to measure the actual gene output of some sensors. We did this by building a number of sensors from scratch. That is, we took the gene sequence, the DNA sequence from the sensor that we identified in the library, had it chemically synthesized by Twist Bioscience, and then cloned it into our fluorescent plasmid. We then measured the dose response curve of the sensor using flow cytometry. Now you can see when we characterize the dose response curves with flow cytometry, the measured gene expression outputs, which are shown in these right plots by these darker points, match pretty well with the predicted gene expression outputs, which are indicated by these shaded regions for all three of these plots. In total, we measured the dose response curves of about 130 sensors using flow cytometry. And the gene output estimates from the growth rate information are accurately representing the function of the sensors in the library. So back to targeting individual parameters. We found that we could identify and build sensors with targeted EC50 values within about 1.25-fold of the targeted value. And we could target gene expression at high gene output levels to about 1.12-fold of the targeted value, although at lower gene expression levels, our accuracy dropped to about 4.3-fold of the targeted value. Now, this may be the first quantification of accuracy for engineering biological function with targeted precise engineering goals. I think that this is overall a good approach to engineering biology. It's an indicator of our true ability to engineer biology in a meaningful manner or with purpose. And I would like to see this happen more often. But possibly more important than identifying sensors in the library that had specific properties was the discovery of rules that may be generalizable to other engineering targets. For example, we found that the effects of amino acid substitutions on the EC50 parameter were log additive. This was seen throughout the entire library. The effect of two different substitutions added to a predictable value. And you can see the predicted EC50 over here against the actual measured EC50 of two different substitutions. These types of rules will greatly facilitate the design and construction of biological systems with predictive and precise functional properties. So at this point, I want to recap before we touch on a few additional benefits of this overall approach to engineering biology. So we demonstrated that we could identify genetic sensors with precisely and quantitatively targeted functional parameters. We measured the dose response curves of over 60,000 genetic sensors and found a wide variety of functional diversity. This allowed us to engineer genetic sensors with a wide range of functions. We quantified the accuracy with which we could engineer genetic sensors with specific properties. This was our overall goal in this endeavor, but we found some interesting things by measuring the dose response curve of every sensor within a library. For example, here in this plot, we see a number of sensors that appear to have inverted dose response curves. They have high gene expression in the absence of ligand, and then low gene expression at saturating ligand. So yeah, these appear to be inverted. And when we built a couple of these sensors and characterized the dose response curves using flow cytometry, we found that the dose response curves of these sensors were indeed inverted from the wild type. This conversion of Lacl from a repressor to a co-repressor has been described previously, but we found a wide range of genetically diverse sensors had this inverted phenotype. And beyond that, they make up a significant portion of the library in total. About 0.35% of the sensors in the library are predicted to have inverted dose response curves. But we found other anomalous functions as well. For example, the growth rate data indicated the existence of band-stop dose response curves. These somewhat strange sensors have high gene expression in the absence of ligand. Gene expression drops at mid concentrations of ligand, and then at saturating ligand, gene expression increases again. We built a couple of these sensors from the library, measured their dose response curves with flow cytometry, and confirmed that this phenotype exists. And again, these are not, they're not super rare events. They're not one of a kind. They make up about 0.18% of the total sensor library that we measured. When we investigated the bandstop sensors within the library, we found one that had only 3 amino acid substitutions. So we know that this novel function is relatively close to the sequence of the wild type sensor. So here on the left side, we see the sigmoidal dose response curve of the wild type Lacl sensor, and we have the dose response curve of the bandstop sensor with 3 substitutions on the right. If we take those 3 amino acid substitutions that exist in this bandstop sensor and then put them into the wild type Lacl sensor and measure their dose response curves, we see that these substitutions don't really have much of an effect on the function of the sensor. They all still have sigmoidal dose response curves. These are the red dose response curves shown in these 3 plots. Although the EC50 of this sensor in particular is shifted to the right. If we take 2 of these substitutions from the bandstop sensor and put them into the wild type sensor, we find that the dose response curves have a little more variation. And in fact, the combination of 2 substitutions results in this bandstop dose response curve phenotype. I find this pretty incredible that there is a bandstop dose response curve, or a sensor with a bandstop dose response curve, only 2 amino acid substitutions away from Lacl, but it hasn't been discovered, especially since Lacl is, as I mentioned before, one of the most studied systems in biology. So, an interesting thing about these inverted and bandstop sensors, as I mentioned before, is that they're quite diverse genetically. So in these middle plots here, the polygon nodes with different sides represent sensors that are either inverted or bandstop sensors. And then the circular nodes that they're attached to represent specific amino acid substitutions in each of those sensors. You can see that the sets of inverted and bandstop dose response curves share very few substitutions. In fact, the diversity of these 2 phenotypes, they appear to be more genetically diverse than the library as a whole. So there's not 1 or 2 substitutions that are responsible for these phenotypes. They are, in fact, very unique genetically. So now we know that the evolutionary landscape of Lacl is filled with functional diversity, and novel phenotypes exist only 2 substitutions away from the wild type sensor. It's easy to conceptualize an evolutionary landscape or a functional landscape that looks kind of like this, where certain mutational paths lead to these new novel phenotypes. And to some extent, that's probably true, given that most amino acid substitutions have a minimal effect on the function of a sensor. But I think it's interesting that these new sensors are so functionally diverse. And in fact, the evolutionary landscape of Lacl is perhaps something more like this, where it's not a slow meandering walk up to a new novel function. Instead, it's quite diverse, and there are many evolutionary paths to all sorts of different functions. This certainly explains why it is so hard to predict or design proteins, given that it is not like a walk up one fitness peak. Okay. So we have an exciting data set here. In total, we've parameterized the function of over 60,000 genetic sensors. We have the exact dose response curve of each one of those sensors, as well as the amino acid substitutions that are associated with each one of these dose response curves. Previously, machine learning methods have been used to predict the function of proteins with moderate success, but those are often predicting unit-dimensional functions. Brighter, faster, tighter. The function of genetic sensors is more complex with multiple functional parameters that have to be considered. But we do have a large data set, and it might be sufficient for training machine learning models capable of predicting the function of genetic sensors. Now, I do want to say this entire portion of this presentation was spearheaded by Peter Toner, who is a postdoc at NIST in the Statistical Engineering Division. He built a deep neural network capable of digesting and processing all of the information that we have, and he was able to reliably predict the function of genetic sensors based on sequence alone. Now, I'm always a little bit dubious when I hear machine learning in engineering biology or anything related to biology. While I think that it's a novel endeavor, and I think that we're on the right track, and eventually we will have computational methods that are able to predict and build complex biological function, I never know how much faith to put in these models. Now, one of the great things about the model that Peter developed was the addition of uncertainties to the model predictions. While usually machine learning predictions have just point estimates, the output of the model Peter developed includes uncertainties, and this allows us to consider each estimate individually based on the confidence of the model. Okay, this model allowed us to determine the effect of single amino acid substitutions on the function of Lacl. So as I mentioned before, 94% of the accessible amino acid substitutions appeared within the library. Overall, about half of them appeared in sensors by themselves. So there was only one substitution in the sensor, which gave us a direct measurement of the effect of that substitution. But the other half appeared only in combination with other substitutions. The DNN model was able to determine the effect of these substitutions, allowing us to collect a more complete set of the effects of single amino acid substitutions on the function of Lacl. Overall, we measured experimentally, there was the effects of 964 different amino acid substitutions, and the model that Peter created predicted the function of an additional 1,027 amino acid substitutions. In total, this allowed us to explore almost 2,000 different amino acid substitutions and their exact effects on the function of Lacl. This accounts for 94% of the total accessible diversity for Lacl. And finally, here is the effect of every individual substitution on the function of Lacl. You can see the effects on 3 different parameters, G0 over here, G infinity in the center, and then EC50 on the right. The results are broken down structurally on the top and sequentially here. Positions that affect a given parameter more than 5-fold are highlighted in either red if they increase the parameter or blue if they decrease the parameter. So you can see effects on G0 always increase G0, and effects on G infinity tend to always decrease G infinity. EC50, on the other hand, can be increased or decreased. And what's interesting is that the effect that a substitution has on the EC50 is related to its position in the structure. Most substitutions that decrease the EC50 occur near the N-terminus of the sequence, which corresponds to this DNA binding domain of the protein. Alternatively, substitutions that decrease G infinity are never within this DNA binding domain. Instead, they're centralized around kind of the center of the core domains, which is near the ligand binding pocket or along the dimer interface. And G0, I mean, substitutions kind of throughout the structure affect G0. So overall, we developed a new method to engineer allosteric transcription factors or genetic sensors by measuring the dose response curves and the exact sequence of a full library of genetic sensors. This required the convergence of laboratory automation, data analysis, long and short read sequencing, and molecular biology. With this measure everything approach, we found a ton of functional diversity. We demonstrated the ability to engineer genetic sensors with precisely and quantitatively targeted functional parameters. And we also discovered new and novel functions that were not previously characterized. We also were able to build a DNN capable of predicting the function of genetic sensors based on the amino acid sequence alone. While this approach should be broadly applicable to engineering genetic sensors, it should be adaptable to many other efforts. Overall, my take-home message is that we should measure everything. With measurements, we can quantitatively characterize biology and the performance of biological parts. This will allow us to, as a field, assemble biological constructs with functions in a predictive manner. And we can continue as a society to improve our understanding of biology. Eventually, we can solve the problems that need to be solved today, and we'll be ready to face the challenges that we will meet tomorrow. So in conclusion, I want to again highlight that a lot of this work that I presented here is in a preprint on bioRxiv, the genotype phenotype of an allosteric protein. If this was interesting to you, we are recruiting postdocs. You can get a hold of me, or you can search for anyone in the cellular engineering group at NIST. I need to give a shout out to Dave Ross, who handled a lot of the work presented here, Peter Toner, who developed the machine learning and deep neural network, and many others who are involved in different parts of this entire process. I believe that is the end. I'm happy to take questions or whatever comes after this.
Thank you very much. That was really awesome. I especially like the conclusion, we should measure everything. I want to measure everything. So you have a few questions in chat. I have some questions too, but maybe go through the questions you got in chat first.
Oh, I can try. I have to say that I have never really used Zoom. How do I get to the chat?
You should see a chat, little kind of bubbly chat icon on the bottom where you see all the other icons like participants, share screen, and all that. You might need to stop sharing screen for that.
Maybe that is it. I'm going to try stop sharing my screen. Ah, there we go, chat. Those two. Those two amino acid substitutions that, yes, so one of those, all right, so did we look at how conserved across lineages, those two amino acids that turn the function to bandstop, did they make contact in the structure? No, they do not make contact in the structure. Although one of them appears in two bandstop sensors, which I believe that's the most represented substitution in the bandstop sensors. Let's see. Oh, I guess one substitution appears 3 times within the bandstop sensors. But one of those 3 substitutions is involved in multiple bandstop sensors. Okay, other than modifying the allosteric protein itself, how would you go forward with fine-tuning the dose response? How would we go about fine-tuning the dose response curves, changing it from a Hill function type on-off to something more linear? And there are a number of examples in the literature doing this. The ones that I'm aware of use multiple sensors or transcription factors kind of stacked together to kind of flatten that dose response curve over a range of ligand concentrations. And that involves changing that N parameter, that 4th parameter that I didn't actually talk about today. That particular parameter of genetic sensor function didn't vary or didn't change very much in the dose response curves that we measured. So it's unclear how malleable that parameter is in Lacl. I'm definitely interested in figuring out how malleable it is across the entire field. I'm not entirely sure. Did you manage to change cooperativity? Oh yeah, again, so cooperativity is also another measure of, or characterization of that N parameter or the steepness. And again, we didn't really see much of a change in the cooperativity. Okay, so I'm curious about whether you think substitutions alter the translation rate or the final Lacl relative expression that may be causing some sort of phenotypes that you're seeing. So we had a number of synonymous nucleotide changes in a bunch of different sensors in the library. So by comparing the function of different sensors that had the same amino acid sequence, but different nucleotide sequences or DNA gene sequences, we did not find a measurable effect in any of those comparisons. So it doesn't look like the DNA sequence has much of an effect on the measurable function of a sensor. And if the population of Lacl had been changing significantly based on those DNA changes, there would have been a measurable change in the dose response curve of those sensors. Now, we did have sensors that had substitutions in the start codon, and those very clearly had reduced concentrations of Lacl, and they have kind of predictable changes to their dose response curve.
So I have a question, not in chat, I'll just do it by mouth. I know the answer probably will be you haven't done it and you think this is insane, but how difficult would it be to do a similar experiment but for a set of genes, like a genetic circuit? You know, I'm asking from the building cells point of view.
Yes, totally. So this is something that I'm very interested in, is using something like this for like metabolic engineering, where you have a string of genes that have related functions. I think this would be a great approach to traditional optimization of those types of pathways or functions, definitely.
And are you there yet? Like, is your throughput enough? Is your sequencing and all that up to it?
We were limited to 10 to the 5th sensors by Pack biosequencing, and I think we could improve that now. Pack biosequencing is continuing to increase their throughput. So I think we can probably get to 10 to the 6th variants right now. I mean, that's a pretty large population to characterize. I don't know what the functional diversity of an entire operon would look like if it was put through something like this. And maybe it would be more, instead of mutagenizing the entire sequence, you kind of swap in libraries of parts where you create diversity in that manner instead, or a combination of the two. Yeah,
if you could do something like, optimize a genome of a cell that's like 100 or 200 genes, that would be really useful, but I imagine difficult.
That is difficult. And I would point you to Sasha Levy at Stanford, who is characterizing the fitness effects of genome mutations using an approach similar to this. But that is much harder. When you have to sequence a full genome rather than a full plasmid, I think your throughput drops.
Yeah. What about not doing sequencing? What about doing something like that with just functional analysis?
Yes, yep, that is definitely a possibility. I think that's yeah, so if we had just done the barcode sequencing instead of the long read sequencing, and that is definitely an approach that is that people do. It does work. And so perhaps something like that would be more appropriate for optimizing more complex systems where you don't necessarily care about everything. You just want to find the best.
Yeah. Okay, that was my practical question. If there are no more questions, thank you very much, Drew, for giving the talk. And if anyone else have any questions, find him over email. And I'll stop recording.
Okay.